Hi there, welcome back to NeuroReview. In this video, we're going to take a quick look at what a neuron is and briefly go over its importance in neuroscience. You might know that all tissues and organs in the body are made up of cells, and that these cells' abilities and interactions with one another define an organ's capabilities. Well, the brain is no different. There are two main types of cells in the brain, neurons and glia. Both are incredibly important in letting the brain do what it does, and there are about 85 billion of each. In this video, we'll focus on neurons and cover glia in a future video. Neurons are the cells in the nervous system responsible for sensing changes in the environment, communicating these changes to other neurons, and coordinating the body's responses to these sensations. This information processing is the key to giving the brain its special abilities that we rely on it for every day. Neurons are very difficult to study. They're about 40 to 200 times smaller than the tip of an unsharpened pencil. Also, the brain has to be cut into very thin slices to study neurons under a microscope, which is hard to do with the brain's jello-like consistency. Scientists had to figure out how to harden or fix tissue and use a special machine to make very thin slices. If that wasn't enough, the brain is largely the same cream color throughout, which makes a lot of the components look the same. Scientists had to develop techniques using various special dyes to stain the different components of the brain different colors, such as the Nissl stain and the Golgi stain. Here's an example of a Golgi stain. It shows that neurons have at least two distinguishable parts, a center part that has the cell nucleus, and many thin structures that extend away from the center part. The center part has many names, the cell body, soma, and perikaryon, all which mean the same thing. The thin extensions are called neurites. There are two types of neurites, axons and dendrites. The cell body usually gives rise to a single axon, which is the same diameter throughout its length, and any branches from it generally extend at right angles. Because axons can extend over great distances in the body, axons were predicted to act like wires that carry neuron output signals. Dendrites, on the other hand, are rarely longer than 2 millimeters. Many dendrites extend from the cell body and generally taper to a fine point. Since dendrites come in contact with many axons, they were identified to act as the antenna of the neuron to receive incoming signals or input. The creator of the Golgi stain thought that his creation showed that neurons were fused together into a giant network, like the arteries and veins that carry blood throughout the body. This idea became known as reticular theory. However, a Spanish histologist and artist named Santiago Ramon y Cajal had a different theory. He learned about Golgi's technique in 1888, and over the following 25 years, he used the stain to map out the circuitry in different parts of the brain. He thought that neurites of different neurons communicated by contact, not by continuity, as Golgi suggested. In that way, Cajal argued that the brain was made up of individual, smaller units. This idea that the brain obeyed cell theory became known as the Neuron doctrine. It wasn't until the 1950s, with the creation of electron microscopes, that the Neuron doctrine could be confirmed. This reconstruction from multiple electron microscope images shows the axon of a neuron in yellow in close contact with, but not fused to, the dendrite of another neuron in blue. To wrap up, in this video we learned that the brain is made up of billions of brain cells, divided into neurons and glia. Neurons send and receive signals in the brain, using many dendrites for receiving signals and a single axon for sending signals, which are both examples of neurites. These structures can be visualized using various methods, including staining techniques and microscopy. These methods helped prove the validity of Santiago Ramon y Cajal's Neuron doctrine over Golgi's reticular theory, showing neurons to be individual units in contact, rather than one big network. In the next video, we'll pick up where we left off and talk about the components that make up a neuron. Thanks for watching this video. If you liked it, make sure to like, share, and subscribe. As always, if you have a suggestion for a topic or a note on something I could improve in a future video, feel free to leave a comment down below. Thanks for stopping by, and I'll see you in the next video.